Yeah, thank you. So yeah, I'm going to tell something about the research that we have been doing in our group and a bit from before that. So if you like my research line that I set up and we're exploring. So bridging chemistry and biology with the resource description framework, a bit about ontologies, cheminformatics, and of course all of this open science. For reference, there is the my Mastodon account here, my ORCID. The DOI of this presentation, it's not there yet, but I will upload that after this meeting, so they can look up details. So our group in Maastricht University, within the group of translational genomics, we study biology and we try to understand the interaction of the chemicals in our body, but also with the chemicals from outside our body, the exposome, so toxic compounds, food ingredients, etc. And what we want to learn and understand is what are the chemical underlying mechanisms behind that. Obvious things from chemistry that we know is drug discovery, medicinal chemistry, toxicology, but also simple things like compounds that are beneficial, like antioxidants, those kind of things. So we have a very broad view of the biology in our department, translational genomics. And what we see here is a biological pathway from our pathway database, Wiki Pathways. This one is created by Denise Slenter. And what we see here is a lot of information. And just even then, just a part of all the biology that is happening in this process. So in blue, we see a number of metabolites. The black boxes, those are enzymes. And then in purple, we see a number of diseases that can happen if something goes wrong in that step in this pathway. So that is some of the information that we want to link together. And we need a lot of information to link together to be able to answer the biological questions that we have. And here I have a compound, and for any compound that we're exposed to, that we eat or breathe in, we want to understand: is this toxic? Is this non-toxic? Is this beneficial for our health? And then, we need to find information about this compound. So how do we do that? Where do we find that information? It's scattered, and all the things that we can solve with the resource description framework, and we have been working on for some time now. But all that knowledge is in a distributed, and there's a very long tail of things that are not in databases. So there's the scientific literature with a lot of information there. It's not easy to get that out. They're written for humans, journal articles, not for something that helps with automation of things. But that's the whole story of there, of course. There is this long tail of knowledge, and this even applies for just the databases. Every year, new databases get created with bits of information that need to be interoperable. Now, one of the things that I started exploring as part of my research is, how can resource description framework help with this, and along with RDF, the various other aspects of the semantic web. And just as in history here a bit, it has been around for some time. So the original specification was published almost 27 years ago. So you would expect it would be we would have some good examples, and I think we do. Some 10 years later, we had a nice symposium in Boston on the use of resource description frameworks in chemistry. We had three sessions there of close to 4 hours, giving a diverse idea of what RDF could do for chemistry specifically. Though most of the research that we do that we do in our group right now is more at the life sciences. It is there. It was a GitHub pages. There is actually RDF in these pages, so we really try to push things. Now a lot has happened since 2010. I'm going to show some of that in the research that we do. So resource description framework was originally in the 99 recommendation. It was based on the extensible markup language XML. And there was some reason for that, and XML was doing a pretty good job in being explicit. Yeah, soon after that, they realized it would actually be nice to be able to serialize information in the resource description framework in other formats as well. So the XML part of that was dropped, and we now have multiple formats. But the underlying structure of RDF is, of course, the triple. It has three components, a subject, a predicate, and an object. And the nice thing about this basic structure of a triple is that it's so enormously powerful. It's so simple. You can model a lot of things with it, and basically anything. It outsources two things that makes that possible. One is you have identifiers for all three concepts. Of course, and we have the literals as well. But otherwise, things have an identifier, and these identifiers, they're unique. They allow us to give more information on it. We can use terms from an ontology, from the vocabulary. And everything is an identifier and becomes self-explainable as long as we can resolve that. The downside of that, perhaps, is it is also a bit more verbose than a lot of formats that do not provide that information, where you just have to guess or make a reasonable assumption. One of the links there is the links to ontologies, so that gives a lot of meaning, a little context to things that help with data curation and what's not. And around the RDF, we have query language, Sparkle. We have languages to express the shape of things like Shex or Shekel. We have ontology-related solutions like the simple knowledge organization system, not really ontology, but towards vocabulary kind of things, and the web ontology language aspects. And we will see a bit of all of that, but together they provide us with a way to be fair with our data, with our knowledge, to such an extent that things become reproducible. And that is really essential, because from a comma-separated values file, it is there. You have to make assumptions. The assumptions are somewhere else. The meaning of the columns, there's even somewhere else. You all have that documented. You will probably do that in a text file. It's not integrated, but with the resource description framework, we have one solution that can basically cover all these things. So here we have some example triples. Here we have that in the turtle format, so not in the RDF XML format. And this is what it looks like. We have subject, etox data set, and then as an object, the void data set, and then some additional triples, the title, description, and publisher, so literal, and a license, again, pointing to an object. And then for a material, we can do the same thing. It can all be in one place. It can be distributed because we have the IRI, so the information about the Creative Commons license, for example, is somewhere else. We can look that up when we want to know more about that. That is an aspect, linked data. We will see that later. The web ontology language, we have a uniform format that allows also things, that allows automation of things. There was other formats, of course, for ontologies, but this one just nicely integrates with the data that we have. So when we have a data set, we can actually load the ontology alongside with that and do an integrated analysis. And because of this automation, we can also much easier make application ontologies, as we see here. So this is a diagram showing the structure of the enhancements ontology, and it actually has components from many other ontologies. As you can see here, from the chemical information ontology, bioassay ontology, Gabi ontology, nanoparticle ontology, etc. So for a certain application, in this case, that was in Nanomapper, the safety of nanomaterials, we can get those bits together. We can do the curation and etc. To really address that problem. And by having all this in RDF, we can document and integrate and everything there, so that we really go towards that much easier, reproducible, and maybe even that AI ready that they sometimes refer to FAIR nowadays. Another advantage and component of the semantic web is the Sparkle query language. And the Sparkle query language itself is not expressed in RDF. Maybe it should have. But at least what we can see here, because everything is basically an identifier, we can interpret this query. We can get more information. We can look up what, and for example, in the third line here in this Sparkle query, in this screenshot, we have an WP protein. Not shown in this screenshot, but we know that protein comes from the WP ontology there, which happens to stand for wiki pathways. And we can look up the meaning. What does this database actually mean with a protein? And that is really important for linking all the data. And I hope to show an example of that a bit later. Another interesting thing here is that the Sparkle query language allows us to really take advantage of that linked data as well. So in line 14 here, we see a reference to a different Sparkle endpoint. So we're combining two different data sources here and running a query on both of them, a federated Sparkle query. Now, RDF is just one representation. As the title of this slide says, there's nothing special about it. It is verbose because so that things are more explicit, easier to reuse, easier to reproduce results done with the RDF. But it doesn't mean that you cannot do that expressiveness, that completeness in other formats as well. Theoretically, you can just use document and comma separated values file equally rich, but you just have to come up with something yourself to do all the documentation because the format doesn't do that and RDF just does. You can map between two files, and one nice example of that is the picture here on the left side. Your spreadsheets can be fair, where a team of researchers from ID Consult they worked out how to make that mapping, and we have a number of different resources or solutions for doing those kind of approaches. The other way around works, of course, as well, very importantly. With Sparkle, we go from a multi-dimensional RDF space, we go to tabular data, because in Sparkle basically always returns in table again, tab separated, comma separated, like data structure for reuse, just with the slices of information that you want. Now, because we can be more expressive, we can address this thing, being precise in your uncertainty. And this was something why I got interested in the RDF when I was doing my multivariate statistics, my chemometrics research, and my cheminformatics research. Because you get a paper, you're trying to understand the methods, you're trying to understand the data that they have there, you cannot find it. And what assumptions did they make? Why did they do that conversion? Some of that may be described in the article, sometimes it's not. It might be explained in code that they do. 20 years ago, code sharing was not very common yet. It's more common now, but even now still not so much. But with RDF, we can really be precise in things. That is also not new. It just gives us the expressiveness to explain things. So if we have look at chemical structures, compound information. We have the smiles as a representation of the structure. There's a lot of information there. No worries, I'm not going to go and suggest to use RDF for chemical structures. That not. But the point is here, we have a chemical structure, and we have information about that. Remember that lookup of that information that we needed for a certain compound we're exposed to. We can normalize that a bit more. We have the INCHI, the IUPAC, International Chemical Identifier. And that makes it a bit easier to compare things. There is some normalization there. Smiles is not unique. Textual string matching thing doesn't work. With the INCHI, that works a bit better, but it has other aspects that doesn't do everything. We will see that with the RDF, we can combine these two, the INCHI, the smiles, with other information into a data structure and actually solve that. And this Colia example that I will go to for chemistry makes heavy use of this. I also want to shout out to the Chemistry Development Kit project that has been running for some 25 years. And that is another important aspect. So RDF is a representation of knowledge, but around that, there is a lot more research that needs to be integrated. And we've been doing over the years some interesting things. So some of the atom types that we use in the Chemistry Development Kit, they're actually expressed in OWL as a data source, and we use that because then we could formalize atom type to atom type mapping. So going from one atom type vocabulary to another, from the CDK atom types, for example, to the Sybil atom types. And in this way, align cheminformatics approaches algorithms in a more explicit way. We can also use the SMILES, the INCHI particular, and other identifiers from a number of databases and make mappings between them. And with RDF approaches, we can express why we say that two things are the same thing. Because entries in DrugBank and HMDB and CABI, they all have a slightly different meaning when they talk about a chemical. And so in a chemical identifier, there is a lot of information there about how we think about the chemistry, something that we will see in chemistry all over the place. The INCHI key, as a derivative of the INCHI, has layers. We can use that. Here's an example from Scolia, actually. And we can see here that the first block of the INCHI is all the same. It describes the connectivity of the atoms and a bit more. And but it excludes information, for example, about isotopes. So we can just do a search on the first block, and in this way get more information than if we just look for the specific identifier. This is particularly clear if we look at the chemical abstracts registry number, the ChemSpider identifier, and the PubChem identifier, which is all different for all the different variants of these of acetic acid. We can also be precise in our uncertainty when we have things like this. Take this example here, this compound. We know the ring structure pretty well, but we don't know where those three hydroxyls are located. We can represent that visually in the diagram, as we can see here. But with the right tools, we can also represent this in a more machine readable way. In this case, in the form of a CX SMILES. And once we have this and we have this annotated, we can start doing processing. We can enumerate all the possible structures here, and then we get specific compounds, and we know that this one is a collection, and we can basically say this is a class of compounds. Something that we do in other places, and that will have identifiers. And this way, we get a much richer but precise network of linked data. We can do that for that compound. We've also been looking at that for lipids, for example. And the bottom left example here, we know the total length, the total number of carbons in the tail, but we do not know where that double bond exactly is located. So we know that Y and X, that those in total combined are 15 carbons, but we don't know if Y is 1 or maybe Y is 10. But in this way, we can represent this kind of uncertainty. A second example in chemistry where we need this is nanomaterials, and that we have had a number of projects in, and we identified there that naming these things is really hard, and using identifiers, they did not actually exist. So what you see in literature, if you look at nanomaterials, they will probably use chemical abstracts identifiers for it, for titanium dioxide or for silica, but it totally ignores the physical chemical properties of that compound. And if you look at the labels that they give them and use in the articles, we see different things, and then you can end up with things like this. So in the top left, we have one silica particle, one material with one set of physical chemical properties, but multiple different names. So we started an identifier there. You can register an identifier, and then we can link information to that. And we've been doing that for in a database. And in this way, we can link information from that long tail. We can mention blog posts, project deliverables, other things that we can take control over with this open science approach. They're not indexed by large databases, but we can do it ourselves. All these technologies are simple and ready. And by expressing that as RDF, we can seamlessly integrate that into our other workflows. So where do we get then? Well, basically the linked data cloud. So this is maybe a figure that you have seen before. This is the whole linked data cloud. This is not just chemistry, not just the life sciences. The chemistry and the life sciences is in here. And if you can read it, you can see the color coding, and the life sciences is pink. That's that big blob that we see in the middle south of this large network. Chemistry here, I believe, doesn't have a particular corner, but Cabby is in the life sciences corner, and Campbell and PubChem and sorts. All these links are the kind of links between record to record, so identifying mappings, an InSheet mapped to a PubChem identifier, as we saw before, and that creates this network. But because also this linked data thing has, hey, we have an article here with a DOI or a PubMed identifier, and it is about a certain chemical. So that is also a link in this thing, and in this way we can aggregate all that information. If we then look at what in our department, we're interested in the kind of databases that we've been using in our integrative systems biology approach. These are some good examples, and these are all RDF-based platforms. Very often, not only RDF, but at least with RDF. So we can integrate them and do federated Spark queries. We can get slices of information from every database, and because we know what slices, we can automate. We can be expressive in integrating all that information, so that we are much more reproducible than what we did basically 25 years ago. In this corner, we contribute to this linked open data network as well, with a number of resources with Wiki Pathways, and you can see the DOIs below the images, the logos here for the adverse outcome pathway wiki that tells us information about the biological responses to toxic compounds. Safe by Design for Nano is towards the same, a similar thing, but then for the nanomaterials. The AOP Wiki database is around the AOP Wiki, but then extended with a lot more chemical information. That is a project that has run on the at the EPA in the United States, and we collaborated with them. Back in the area that I mentioned, 2010, around that time, I started playing with creating RDF for the Campbell database as well, and we have some papers around that, and this is one of them there in that DOI. For the ontologies, I mentioned the ontologies already. These were developed in collaborations before, and Jenna Hastings, first author on both of these articles at the Cabby team in Hingston at the time. But we're maintaining these right now. So these actually have been running, used for quite some time already, but still need a lot of work, and we're trying to contribute to that. Fortunately, particularly in NFDI for Chem, the ontology work is really being picked up. The role is being seen there, and I'm really happy about that. And last year we were happy to join the workshop by the NFDI community. So where does that get us? So we have linked data that can help us understand the biology of these processes, and possibly the individual diseases here. But that requires that for each of those resources that we may want to integrate, we need to be able to understand how do we integrate them. So we need things documented, and some new developments over the last 2 years via biohackathons and the semantic web and applications conferences. There has been some collaborations between a number of these semantic web projects in the life sciences and leaning towards the chemistry. on doing this. And one of that is this the software. I think it originated from Jerven Bolleman at the SIB, where we can document Sparkle, individual Sparkle queries. We can give the documentation. It will automatically visualize the query here so that you can see it. But it will also give information like who authored this, not this particular one, but we can add that. We can even give a DOI to an individual Sparkle query so that they become citable in a data site-like approach. But what this documentation also does is actually what Sparkle query can you run this against? And that basically then acts as a bit of documentation for what is in that Sparkle endpoint. Same kind of thing we've been doing in a different project. This is used in a course that we're giving a practical programming in the life sciences. And here the students use Sparkle, they learn to use Sparkle, they learn to visualize data that they get out of the Sparkle endpoint in a web page. But for that too, they need to know what is in the Sparkle endpoint. So the documentation there too is important. And this resource, we started out documenting that for life sciences databases. And where possible, include that diagram. Like we see there with what entities, is it just chemicals in there? Is there other things like key events here? What links are there to other databases? And you can see the cheminformatics ontology and some other ontologies being used here as well. Unfortunately, such schema are not always available or not easily found. So over the last 15, 20 years, there has been a lot of work done on dynamically discovering what is in a Sparkle endpoint or what is in an RDF data set. And since the point was, when I stressed it at the start, it is much more self-reproducible because by being more explicit, we can actually automate this. And that turns out a bit harder than we hoped because ontologies are not automatically always loaded in the same Sparkle endpoint and those kind of things. And when they are, you get so much information. How do you select just the bits that we have to look at? And this is a project from one of the PhD candidates in our group, Javier, who's been looking at this, and down there you can see his ongoing project right now in helping to document that. And one of the things that that tool does, it looks at all the data, it tries to find the patterns there, and for some of that, it really works quite nicely there, and it will give you a diagram like this. And you will actually take this a step further because what is behind that, what we need for answering the biological questions that we have and so on, we kind of need, hey, we have this disease here. We know the genes that are metabolites that are involved. Which database actually can help us here? Which of the long tail of databases, of the hundreds of databases that are around or have been published in the last 5 to 10 years can help us there? We need a dynamic discovery. It is basically the semantic web upscaled version of, hey, let's do a keyword search in PubMed. Then back to Wiki Pathways. We have that information. We have the links out. So we had a good start already. That really helped us. Integrating this information. The semantic web allows us to do that integration, linking out to databases, but also the data curation. So those curation badges that you can see here is based on the RDF because the RDF allows us to find contradictions in an automated way, not just within Wiki Pathways, but also by integrating other knowledge bases. Because it's so reproducible, we can present this to the curators of the database as well as the readers. Here we have one of the pathways. There are literature references to them, so it's not just even you don't see the literature references here, but they can be on individual data nodes, on the individual metabolites, or on the interactions between them. And because we can then use the RDF to extract information from about the metadata from those articles and sorts, we can actually track what literature has been published in the last few years about this type of interaction. Get that from a literature database or perhaps from databases like Uniprot. where they're very actively tracking literature about, for example, enzyme activity. So that's this is just a screenshot of showing the linking out and the citation JS for the references. Here is an example. We now only track the identifier for the article, whereas in the past we would have typed the whole article or copied the whole article title. We just have the identifier, and the process of creating the website just pulls it in a semantic way. Now, the last thing that I want to show is Scolia, where we very actively use this as well. Scolia is based on Wikidata, and here we see a screenshot of Wikidata and the kind of information that we have there. This screenshot comes from a 2021 article, and we see some information here. We have the same kind of structure in Wikidata. We have a subject, which is retinoic acid receptor alpha. It has a molecular function. That is sort of the predicate. And then we have as an object here highlighted at the top, retinoic acid binding. Wikidata effectively is a large database with a lot of information from many different domains, and all this is here depicted as statements in the interface, but is also available as RDF. And we've been using that for writing Sparkle queries against this database, and for the life sciences, but also for chemistry. And that is that poster that you can see the top right, now available as a preprint. It started out as very broadly in the top left, enabling open science. There was a grant proposal that we made to the European Commission. Unfortunately, not granted, but we took things forward just at a slower pace. And that led to a lot of things. Scolia, that is the middle top, more or less. We started using it for the life sciences in there. I connected with a former colleague, Andra Waagmeester, who had been working with a team in San Diego, with Andrew Su on doing a lot of information in Wikidata. And during the corona pandemic, we actually looked at enriching Wikidata with coronaviruses because we had the SARS-CoV-2 virus pandemic, and we did not have a knowledge base for that. We did not have identifiers for the proteins in the coronaviruses. So we had to mint them, we had to create them ourselves, and we used Wikidata for that. And the other two is a collaboration with Chemical Abstracts and with the complex portal database at the EPI, linking all this information via Wikidata so that it is available. Now we can, because there is so much information in Scolia, in Wikidata, we can visualize that with Scolia and in graphical ways, as we can see here on the left side, and that is a number of topics related to a set of articles. We can look at a single article there, have the information there. And then a bubble plot that we see on the left side in Scolia typically is based on the main topics of that particular article, but it actually knows also which articles are being cited by this article and which articles are citing this article. And those topics probably have something to do with this article as well. So they can be shown in the bubble plot as well, but with a lower weight, they're smaller. But in this way, you can recognize patterns. Same way, you can find related articles. You can list the citations to the article, and if you sort that by the number of citations that they get, it can point you to interesting new articles, or articles that may have information. Some are hidden in that long tail, relevant to the biology or chemistry that you're interested in. We can do that for classes of compounds, and then we can make a plot as we see in the top left there. We can do that Inchiki thing that I showed earlier. Because we have the identifiers there, you can actually look up that identifier and get information about that compound using that identifier in the top right. We have the chemical structure there. This is a nice visualization that I like. It's based on the smiles in the isomeric smiles in Wikidata, but we have the R and S annotation there for free, so we can really see what the chemical nature of that chemical. So that sort of wraps up what I wanted to show, but there are a couple of things that I really hope you will take home. And some of that we see in a number of projects already. It's really not unique here. But for our research, in particular the metabolomics, we need cheminformatics, we need knowledge graph, we need that combination. And we have a growing number of solutions and, if you like, standard operating procedures, methods implemented as software that we can do to do all these things. Compared to when I did my PhD around 20 years ago, I was still working on it at the time. We can be precise in our uncertainties yet. And somewhere in one of these publications one of the studies that we did, we showed that this actually helps the machine learning. The linked data article from around 2012, around that time, we showed by using by being able to use this uncertainty, but because it's semantic in an automatic way, we can actually make our predictive models more precise as well. We know the uncertainty, we can compensate for that. And since then we've been extending this and bridging things. And here things are really important because the chemistry really is differently described in different databases. And chemical entity in Cabby is really different from a chemical entity in for example, the human metabolome database. One has a more biological focus, the other a more chemistry focus. They link to each other, but they're not identical, despite they have an identifying mapping database. And this shows up a lot of times, also in Wikidata. There are a lot of reasons why things can be the same or not the same. Bridging knowledge is not easy, but we can be precise in it now. Despite this, and that linked data network is really getting pretty large. The number of even SPARQL endpoints where you really have the very detailed access is growing very nicely, and there is more that we can do right now than we have time for right now. But there is still also so much that is not fair enough in doing these things. I just hope to have convinced you that starting with an RDF is definitely a good way to do. So we should do that more. It's much easier to go from RDF to a table than from a table to RDF. And what I just said, the open science, the number of tools that we have, the amount of knowledge and data that we have in a sufficiently RDFized way, fair enough, and the open licenses make so much possible. And we've been using that very happily over the last couple of years. So that wraps up my presentation. So this is the opening slide. I just want to thank the funders at the bottom that have funded a number of projects where we've been using these approaches, starting from Mapper on the right side, number of other nanosafety, a nanosafety cluster project, now also PHP for safety, but also funding for the capacity development kit for BridgeDB, for Wiki Pathways, and other resources there. And a specific shout out to the team that are actually doing all this RDF work in our group right now. There's Javier, Sidra, Denise, Marvin, and Ozan. And that's from left to right, and at the far right, there's Tuba. She was doing integration in the semantic approach as well with biodata views in our team, but she recently left for another position at Amsterdam. And all that together with a lot of people. So there is a lot of very long list of co-authors there that I also acknowledge because I really like this of the open science approaches, all the collaborations, all the shared ideas and making things possible. Thank you.